How's it going, everyone? It's Chow here, and today we are going to talk a little bit about the eye and the phototransduction cascade. So let's get started. Okay, so first of all, we have vision. Now, vision is basically just conscious sensory information that we take up from the eye, pass it through into our central nervous system. That's basically what vision is. Now, when it comes to human vision, we have the eye, which is fairly important, obviously, but there are different components of the eye that will help us see the world around us, at least get it initially started. So first of all, we have the light that's coming in, and it's going to strike our cornea. Once it passes through our cornea, it's going to hit our pupil, and this pupil can dilate, obviously, to basically let in light, and depending on how much light is available, it might be contracted or it might be dilated. We then hit the lens, which allows the light to be focused onto the back of our retina. And in the retina, there actually are a bunch of different cells that basically signal, and that's where the phototransduction signal cascade kind of initiates. And those signals, those action potentials, will eventually be collected at the ganglion cells, passed into the optic nerve, and then eventually it's going to be passed into the brain. So information from the eyes are going to be processed in the brain at the visual cortex, at the back of the head. So again, remember, think back and try to get that idea that the occipital lobe is where most of our visual processes are going to be, well, processed. So that's vision in a nutshell. Now, if we look at the retina, so this little thing back here where all of our cells are, the retina is basically made of different cells that innervate together. One of the most important cells is this group of cells called the photoreceptors. So these are your photoreceptors over here. The photoreceptors are going to basically be connected to the bipolar cells, and the bipolar cells are going to then innervate into the ganglion cells over here. The ganglion cells will eventually collect to form the optic nerve, which will be passed into the central nervous system. Now, one thing to keep in mind is that in terms of the direction of light, while indeed the photoreceptors are back here, the direction of light in this image is actually from right to left. So that's something definitely to keep in mind. And you could perhaps think about it how the light actually technically strikes the ganglion cells first before it actually reaches the photoreceptors, but in order for the transduction cascade to actually begin, it starts at the photoreceptors. So the light direction in this particular image would be from right to left. Now, photoreceptors themselves are very important because they're what get the whole transduction thing started. So there are two different types of photoreceptors, but the photoreceptors themselves are actually metabotropic sensory receptors. So they're transforming that light energy into an action potential. The two types of photoreceptors are rods, which are sensitive to light, and cones, which are sensitive to basically color. So if you look at this diagram of the eye over here, you can see that the light is passing, in this case, from left to right. And it's passing from this way to that way. But if you look at the diagram of the retina over here, you can see that the cone cells and rod cells, the photoreceptors, are actually at the very back, whereas the ganglion cells are over here. So the light actually passes through, hits the photoreceptors, they pick them up, and then the transduction cascade starts, and the action potential actually goes backwards. So that's something to definitely keep in mind for your exam. Now, in terms of the actual phototransduction cascade, there are a number of steps that occur. So let's just go through this, and hopefully at the end of this video you have a better understanding of what's going on. So let's use a rod cell, for instance, which is going to be very sensitive to light. So we have light that's going to strike the rod cell. It's going to go through the outer segment of the segment membrane of the rod cell to the inside. Inside, there is this protein. So there's this protein called opsin. And inside the opsin, there's going to be this other molecule called retinal. So light is going to strike the opsin. So you can have a photon of light that comes in and strikes this opsin molecule. This opsin molecule inside, again, has this sort of molecule called retinal. Normally, it's in the 11-cis retinal conformation. But when light strikes this opsin, that 11-cis retinal changes to an all-trans retinal. In addition, the opsin itself also kind of shifts in its shape. This causes the activation of a G protein called transducin. Transducin takes GTP and hydrolyzes to form GDP, so it's using a hydrolysis reaction to gain energy. The activation of the G protein also causes the activation of PDE, or phosphodiesterase. Now, this phosphodiesterase, what happens is it actually breaks down cGMP, or cyclic GMP, and turn it into normal GMP. Now, cyclic GMP is very important because normally cyclic GMP is bound to this sodium channel over here, and it actually keeps that sodium channel open and allows for an influx of sodium, which causes depolarization. However, when light strikes, right, it's going to cause this cascade that causes this phosphodiesterase to change the cGMP into normal GMP, and so they are no longer going to be bound to the sodium channel because you're removing that cGMP and it no longer is available to bind to those sodium channels, and those sodium channels end up being closed. The closing of the sodium channels means less sodium entering the cell, and this causes hyperpolarization. Once the cell is hyperpolarized, once the rod cell is hyperpolarized, it actually no longer is able to inhibit the bipolar cell. So remember, if you think back, you have our photoreceptors and then we have our bipolar cells over here. Normally, the bipolar cells are inhibited, but when light strikes the photoreceptors, the bipolar cells are no longer inhibited. So we lose the inhibition on the bipolar cells. When that happens, the bipolar cell is turned on. So now we turn on the bipolar cell with that light striking. After the bipolar cell is turned on, they fire action potentials into the ganglion cells. So it fires action potentials into the ganglia. Again, the ganglia eventually collect into the optic nerve, and then they transfer that information, that action potential, all the way to the brain. And that's how we basically perceive, oh, we're seeing light. So this is the phototransduction cascade, starting with just a photon of light, or maybe more than a photon of light, striking the opsin. The opsin inside has a retinal molecule that's in the originally in the 11-cis formation. It's going to change into the all-trans formation, so changes from 11-cis retinal to all-trans retinal. That's going to cause an activation of a G protein called transducin, which is going to activate a phosphodiesterase, which is going to break down cyclic GMP to normal GMP. It's going to shut off these sodium channels, cause hyperpolarization, and then ultimately trigger the bipolar cells to fire action potentials into the ganglia, which eventually collect into the optic nerve and then is sent to the brain, and then that's how we perceive, oh, we're seeing light. So again, phototransduction cascade, as well as some basic information about the light. I hope you found this useful. Best of luck studying, and as always, I will see you in the next video.